Good evening and welcome to the December 2022 meeting of the Linnaean Society of New York. I'm your president, Rochelle Thomas, and will now call the meeting to order. While the winter holidays are upon us, I'm already looking forward to spring and to our annual meeting in March in particular, which will be held in person at the Liederkranz Club for the first time since 2020. I hope you will save the date, March 14th, when we celebrate this year's Eisman medalist, Dr. Scott Edwards. As a reminder, the annual meeting is open to members and their guests only. So if you have neglected to renew your membership or are interested in joining, now is the time to do so. Now I have the sad job of announcing the passing of two society members, Emily Payton and Louise Frazier, both active members of the birding and naturalist community here in New York and a friend to many in tonight's audience and beyond. As always, tonight we've disabled the chat feature, so if you have a question, please type it into the Q&A box. Our Vice President, Gabriel Willow, who is broadcasting live from a mysterious destination somewhere around the world, will answer your questions at the conclusion of the lecture. On to business. The votes are in. Motion number 1, to accept the following members of the Linnaean Society passed with 145 in favor and 1 opposed. New members and sponsors are Loianne Beausoleil, sponsored by Bruce Yolton, Catherine Figueroa, sponsored by Chuck McAlexander, Sue Bernstein, sponsored by Ken Chea, Karen Sosnitsky, sponsored by Leslie Fisk, Meredith Barchat, sponsored by Leslie Fisk, Marcy Einhorn, sponsored by Ann Lazarus. Welcome to our newly elected members. Motion number 2, to accept the November 2022 meeting minutes, passed with 145 in favor and 5 abstentions. And now I have the honor of introducing Dr. Jessica Ware. Dr. Jessica Ware is an associate curator in invertebrate zoology at the American Museum of Natural History. Dr. Ware's research focuses on the evolution of behavioral and physiological adaptations in insects, with an emphasis on how these occur in Odonata and Dictyoptera. She holds a bachelor's from the University of British Columbia in Canada and a PhD from Rutgers. Dr. Ware is the past president of the Worldwide Dragonfly Association and serves as a current president for the Entomological Society of America. She was recently awarded a Presidential Early Career Award for Scientists and Engineers Medal from the U.S. government for her work on insect evolution. Tonight, Dr. Ware will present insect evolution with an emphasis on dragonflies. Thank you, Dr. Ware.
Thank you so much for having me. Let me get my slides started here. Can you see the slides okay?
Okay, yeah, perfect. They're great. Perfect. Yeah.
So thank you very much for having me. I'm excited to be here. I too am looking forward to the spring, although, as we'll talk about a little in just a few moments, there are still some cool dragonflies that you can collect even in the winter, because many of the nymphs that are they develop in freshwater, you can still collect them year-round in certain parts of the world, and in other parts, people even take ice cores. So I suppose even in the winter, one can still work with dragonflies and damselflies. So I'm a curator at the American Museum of Natural History. I study evolution, and I'm a field biologist. I travel to different parts of the globe to collect the insect species that I'm interested in using so that I could look at their genetics, their genomics, or their phenotype, their morphology. And in doing so, I'm always kind of amazed by the sheer numbers of insects that are out there. And many of us have a favorite insect group that we know backwards and forwards. And the insect group that I am most familiar with are the dragonflies and damselflies, although I also work on termites and cockroaches and a few other insect orders. And there's around the same number of species of dragonflies and damselflies as there are mammals, around 6,500 species or so. We have 1.5 million species of insects that have been described, but many people think that there's 3 million or 5 million. And some people have even had kind of fantastically higher estimates as well for how many millions of species of insects that are yet to be described. And when we look across this diversity of insects, what we see, just as when we look at any of the organisms on the planet, is that there's a lot of heterogeneity, there's a lot of variety. There are things that are herbivores, things that are blood feeders, things that are detritivores, things that are voracious predators like dragonflies and damselflies are. size, shape, form, function. All of this really varies dramatically. And we can see how different body forms have been selected for. Of course, these are all the phenotype. And the phenotype is really determined by the genotype or the genetic information. And that's a lot of the data that we look at in my lab. We look at both genotype and phenotype. So as I mentioned, I mostly work on Odonata, which are the dragonflies and damselflies, but I also study the Blattodea, which are the termites and cockroaches. Termites and cockroaches comprise today a single order, the Blattodea. Since about 2007 or so, we've considered termites to just be fancy social cockroaches, and they're nested deep within the tree of life of cockroaches. The types of questions that we mostly are interested in answering are kind of basic, fundamental who, what, where, when, why type questions. So I'm interested in understanding how different insect groups are related to each other. And then once you have a phylogeny, it really opens kind of a whole bunch of doors in terms of the types of questions you can pose. So I want to know, once I have a phylogeny for whatever insect group it is that I'm working on, I can then ask questions about the evolution of particular characters. I can ask questions about when particular groups first arose on the planet, and what some of the drivers might have been for diversification. Because although there are a lot of species of insects on Earth, not all groups are really species rich. And even within the Odonata, or the dragonflies and damselflies, there are some groups that have a ton of species and others that are relatively species poor. So understanding that is really, to understand that, one really needs a good phylogeny. So I'm pretty fortunate because I originally am from Canada, and I had almost unlimited access to green space. These are some photos that were taken at my Nana's house. She's 97 now, and she lives in Northern Ontario. And we basically had the run of her kind of backyard and her outdoors. And so my association with insects was not with pest insects. My association with insects were things that were usually around water, mayflies, stoneflies, caddisflies, and of course, dragonflies. These were things that we saw pretty much every day. So this is what a phylogeny of the insects looks like. This is a phylogeny that colleagues and I made using transcriptomes, which is the part of the genome that's the genes that are being transcribed, that are being actively made copies of at the time of when we caught these insects. I like to think of the genome kind of like a recipe book or like a cookbook. When you go to make supper at night, you don't make every single recipe in your cookbook, right? That would be bananas. That's too much. You only make the recipes that you're actually going to be cooking and using for that evening. And our genome is kind of the same. So most of the genome is wrapped up in these little spools called histones, and only the genes that are in use at a given moment are the ones that are actively being copied. So when we sequence transcriptomes, we're just kind of sequencing those genes that are actively being copied. And so in this phylogeny, we use transcriptomes to come up with this kind of first tree of insects. It had 144 species in it. And we have a new tree that we're working on right now that has over 2,500 different species of insects for which we have transcriptomes. I thought I would show this as kind of a map, just to overview what we know about the insect systematics, or the insect tree of life. So at the base of the insect tree of life are what we call basal hexapods. These are things like silverfish or firebrats or springtails. These don't have wings, and they tend to live in and amongst the soil. They're sister to the group of insects that does have wings, which are called the pterygoda. And the pterygoda we divide up into two groups: the non-holometabolous insects. These include things like dragonflies and damselflies, cicadas, earwigs. These are things that don't have complete metamorphosis. So they have a juvenile stage, and the juvenile stage often looks an awful lot like the adult, just smaller, and they molt up to this adult stage. Then there are the insects that do have complete metamorphosis. These are called the holometabola. These are things like caterpillars that go from an egg to a larva, or the caterpillar, to a pupa, where there's a complete rearrangement of their internal anatomy, to a butterfly. So there's this pupal stage is what kind of defines the holometabola. So where dragonflies and damselflies are right here at the base of the winged insects. So we think they're among the earliest branching lineages within winged insects. And it's a really great part of the tree to study because there's a lot of interesting questions one can pose. So we think that they're relatively ancient. There are close relatives that are not dragonflies. They're griffin flies that flew during the Carboniferous. They had really big wingspans, about 37 centimeters for each wing, so about 70 centimeters total. They had a beak. They were predators. Most modern dragonflies and damselflies, we have fossils from around 250 million years, depending on who you talk to about the fossils. Some of them never travel far beyond the pond or lake from which they emerge. Some records of individuals travel maybe 11 meters their entire lifetime. And there are others that are really long distance migrators. So this is a great group to kind of study how dispersal can lead to variation among populations and among species. The juvenile stages, even though I mentioned that non-wholly metabolous insects, the juveniles look an awful lot like an adult, that's not the case for odonates because the juveniles are aquatic. So females lay their eggs in fresh water and the nymphs develop in fresh water and then they emerge as an adult dragonfly. Whether in the nymph stage or in the adult stage, they're voracious predators. So they're really good at controlling populations of mosquitoes, of black flies, of horseflies. They also eat each other and pretty much anything that flies. We think that they were among the first things to fly. So before birds, bats, or pterosaurs, insects were the first things in the sky, and probably it was something that looked a little bit like a dragonfly or a damselfly. They have a global distribution, and one of the things that humans have noticed about them and that we've been fascinated about for a long time is that they're very colorful. So they come in a variety of colors: purples, blues, greens, pinks, yellows, and what have you. And part of that is used for thermoregulation. Part of it is used for crypsis or camouflage, but part of it is also for signaling. And for the purposes of being highly visual predators, but also for the signaling that males signal to females and males signal to other males, dragonflies and damselflies actually have excellent vision. So we could study their vision as well. So we divide this order up into three extant or living suborders. There are the dragonflies or the Anisoptera. These tend to hold their wings out to the side, and they tend to have a slightly thicker abdomen. And then there are the damselflies or Zygoptera. These tend to hold their wings behind their back, and they tend to have a very slender abdomen. There's around 3,000 species of each of those suborders. And like I mentioned, this is around the same number of species in total for this order as there are for all of mammals. And then there's this third suborder, which exists today only in China, the Himalayas, and Japan. It's called Anisozygoptera. It exists today as a single genus, Epiphyllevia. And that genus had 4 species, but I'll put a little asterisk there, and I'll say more about whether we still think that's true in just a moment. So this is what the juvenile stage looks like. They have a labial mask or a mouth part that kind of could shoot outwards and can grab prey items and bring the prey items back towards the inner part of the mouth where the mandibles are so that they can consume their food. As juveniles, they mostly are also eating aquatic insect larvae like chironomids and other types of flies, but they can take vertebrates too. So they can take small, minnows, and they can take tadpoles. So in terms of what's actually happening at the base of the tree, there have been several hypotheses that have been suggested. One is that perhaps mayflies and dragonflies are sister to each other. They share a common ancestor, and that is sister to the rest of the winged insects. Another hypothesis is perhaps that dragonflies were the first thing to kind of branch in the winged insect tree of life. And then the last possible scenario would be that mayflies were the first. For a lot of us who study dragonflies, we've favored hypothesis B, which we call the Chi-star-Mararia hypothesis. It's a good Scrabble word, because dragonflies actually do something that a lot of other insects don't. So what dragonflies do is they do indirect sperm transfer by having 2 penises. So when we think about what was flying around the time of dinosaurs, when we think about what was flying in these earliest skies, many of us think that it was dragonflies and damselflies for this reason. This is what the secondary penis looks like. Males have a traditional penis at the tip of their abdomen from which they ejaculate sperm, but then they have a secondary penis at the base of their abdomen, kind of near where their navel would be if you're imagining it relative to a human body. This is what in the upper left-hand photo is just a light microscope photo of what the secondary penis looks like. And the remaining photos on this slide are all of scanning electron microscope photos of what the secondary penis looks like. This secondary penis, they transfer sperm from the primary penis, put it into the secondary penis, and then the sperm is indirectly then transferred to the female. Males clasp the female behind the back of her head, and if she is receptive, in some cases for damselflies, it's actually like a lock and key mechanism where only a certain lock can fit into only a certain key, which is a way to kind of maintain species boundaries. Not all dragonflies and damselflies have that. Females will bring her abdomen up, and then that's when the sperm transfer occurs. But what's neat is that the secondary penis also does this other function because females can mate multiple times, and she can store sperm. She has two types of sperm storage organs. She has long-term storage and short-term storage. The bursocopulatrix and the spermatheca. The spermatheca is the long-term storage. The bursocopulatrix is the short-term storage. Males want to ensure paternity, and so males have actually had selection act on this secondary penis to also do this function of sperm displacement. So many times for when we look at species, what we see is that the secondary penis actually has almost like a spoon or a scoop, and sometimes it looks like claws, and the males actually scrape out the previous male sperm before depositing their sperm, again, presumably to ensure paternity. There is still some female choice because we don't exactly know how she does this and what she's using, what cues she's using to make these decisions, but females store some sperm in the long-term storage and some sperm in the short-term storage. We know that she can choose which sperm she uses from these different storage organs to fertilize her eggs. And we also know from a very small number of species that she can destroy some of the sperm that's in her sperm storage organ. And how she does that, we really don't know. So when you see males and females together, they make this copulatory wheel, and the majority of the time that they're together is actually that sperm displacement step. So I've been really interested in my lab in understanding the way that females lay their eggs. All damselflies and some dragonflies lay their eggs by using an ovipositor. An ovipositor is an egg-laying apparatus that a lot of insects have. In things like bees, ants, and wasps that have stingers, that's actually the stinger is a modified ovipositor. In dragonflies that have them, and all the damselflies, the ovipositor are these three little sclerotized or hardened structures that fit together like a little knife, and they cut holes in plant material and they deposit their eggs in plant material. We call it endophytic overposition because it's within plants that they're laying their eggs. To do this, they need time because they have to cut a hole in plant material and then put their eggs in one at a time, which makes them very vulnerable to predators like frogs, and birds and lizards that are always on the prowl for these as a tasty treat. But it also requires that there's emergent vegetation in the water in which they're laying their eggs. So by contrast, there are two groups of dragonflies, the Libelluloidea and the Gauntlets, which are two taxonomic groups. Both of those have lost their ovipositor. So those three hardened little structures, that little knife, is gone, and instead they squirt out their eggs in a clump and tap their abdomen on the surface of the water. It's very fast, so some have argued that speed might allow them to more quickly evade predators, but also it allows them to kind of expand their larval niche space because they can take advantage of water that pools up after a rain, so temporary or ephemeral transient bodies of water that maybe are too small or too they dry up too quickly for there to be emergent vegetation. They could still lay their eggs in that water. So we have questions about the number of times this ovipositor or this egg-laying apparatus has been lost. We need a phylogeny for that. So this is what the dragonflies look like when they do this behavior, where they've lost their ovipositor. They're just tapping their abdomen on the surface of the water, and the female just kind of disperses her eggs in these giant clumps. And you can see it's very, very fast compared to the time it would take to use this egg-laying apparatus, this little knife, and cut a hole in plant material. Very time-consuming. So to get at questions about like this, like how do these these behaviors evolve, we need to understand how these taxa are related to each other. We need to know which lineages arose first, how the families are related to each other, and that can then give us the opportunity to ask these questions. Traditionally, a lot of dragonfly and damselfly systematics or tree making was based on wingvenation. Wingvenation is a very visible, very easy to score character. But it turns out, although there are variations among families in the types of patterns that you see in wing veins, they're actually really strongly correlated with flight, because more dense wingvenation makes for a stiffer wing, and sparser wingvenation makes for a more bendable wing. So depending on if a dragonfly or damselfly is doing longer flights, gliding, or flying higher up in the air column, they might want to have a stiffer wing versus flying in and amongst vegetation or maneuvering. They kind of is a trade-off between gliding efficiency and maneuverability. We've also noticed that there's a lot of variation in color. Color was a character that people sometimes would use in the past. What we now think is that color actually is maybe a really good indicator of some of the diversification processes that we find in dragonflies and damselflies. So too is dispersal ability. So the two groups, when we look at the groups of dragonflies and damselflies that have the highest number of species, it's groups that have the highest amount of color. The most colorful families are also the most species-rich families. And also the families that have the most variety of flight styles also are the most species-rich. So we have a grant right now where we're kind of testing hypotheses using phylogenies about dispersal ability and color and how they might be related to the evolution of dragonflies and damselflies. But anyways, I'll go back to our discussion. So we need to have a phylogeny so that we can answer questions like whether or not this egg-laying apparatus was lost multiple times. Historically, there were several difficult nodes that were kind of hard to resolve. So people tried morphology, people tried sequencing genes by genes in the 90s and the early 2000s. And it seemed like no matter the lab, no matter the data set, we still had some nodes that we just couldn't seem to find an answer as to who was related to whom. One possible, I'm just going to talk about dragonflies or the Anisoptera, and one hypothesis was that the Aeshnidae, this is an example that's shown here, that these were actually sister to all of the rest of the dragonflies. These are kind of well known. You may have seen them before, especially if you're in the New York, New Jersey area, because in the early to mid 2000s, my former PhD advisor and some colleagues at Princeton actually glued radio transmitters on some Aeshnids and kind of tracked their migration, following them in a helicopter, which was a pretty neat study. And we think that most of the studies that we find, we think that they are indeed sister to the rest of all of the dragonflies and damselflies. But then who falls after that in the tree? Some have said that it's petal tails or the Petalluridae. These are a group of dragonflies that are actually very species poor. There's only a handful of species in this family. And the one person who kind of devoted their life to studying this group was this man named Perry Turner. And while he was doing his PhD work in the Pacific Northwest, he became convinced that perhaps this was the primary diet of Sasquatch, and that Sasquatch were rare in the habitat because petal tails were rare in the habitat. And in this kind of subsequent follow-up to his thesis, he actually wrote that if one were to eat a petal tail nymph, one would turn into a Sasquatch. He actually disappeared. It's kind of sad, but nobody has seen or heard from him in about 20 years now, I guess 15, 20 years. But this is kind of forever now associated with this family as this kind of lore of Sasquatch. But where the position of the petaller is, which I've kind of outlined in this purple color here, where they fall in the tree, we really don't know. We've often found them as sister to the Gauntlets, which I've colored in green here. The Gauntlets are the clubtails. These are dragonflies. The tip of their abdomen, you can see it kind of is sometimes expanded. So it looks a little bit like a club. A lot of people who are dragonfly aficionados love Gauntlets, and they have I think it's the most collected group of dragonflies. People think that they're very beautiful. They often have very bright colors, and this club tail can really vary among species, and they're real collector's items. So we can use a phylogeny to kind of tease apart how these different families are related to each other. And And so this is an example of a phylogeny using close to 500 genes that we reconstructed for around 136 dragonflies and damselflies. We've done similar work with transcriptomes. Again, transcriptomes are when you have all of the genes that are being actively kind of copied at the time when you catch the insect. And in this case, we have close to 3,000 or so genes. And when we put these kind of different data sets, slightly different taxon sample, we put them side by side, what we see is a lot of congruence. So I think what we're converging on is probably the true tree of life for dragonflies and damselflies. Different labs, different taxon sample, different data sets, kind of all of us who are working in this area, we're all kind of coming together to find the same relative answer, which is an exciting time. So I'll just give you a brief overview. We find that the damselflies, when we do divergence time estimation, which is basically where you look at the genetic information that you have mixed with fossil data, you can figure out the timing of when a particular group emerged. And we find that the age of damselflies, or the zygopter, is around 225 million years old. If we zoom in, one of the hypotheses was perhaps that this Hemiphlebeidae and Lestoidia, that they were the sister to the rest of the damselflies, and people had written about that in the literature quite a bit, and we recovered that to be true. This third suborder I mentioned that exists today only in China, the Himalayas, and Japan. It used to have 4 species, but spoiler alert, we just sunk one of them, so now there's only 3. We've recovered a sister to all of the dragonflies. This is a group that's a natural grouping called the Epiprocta that people had found in the past. You can look at this inset map. They have a really unusual distribution. So China, the Himalayas, and Japan, maybe it's just one species with isolated populations. There's a lot more work still to be done on this group, and there's really very few people working on it. So if you like dragonflies and you like a mystery, Epiphlebeidae or Anisozygoptera could be a really great group. Please join us in this work. For the dragonflies, we also recover them to be around 225 million years old. And the reason why I'm going to zoom into this part of the tree is because I had this question, right, about whether or not the ovipositor or the egg-laying apparatus, whether it was lost once in the Gauntlets and then once again in the Libelluloidea, or whether they share a common ancestor, and perhaps it was just, a single loss of this. Well, if we look at it, at where these things fall in the tree, the Gauntlets is actually recovered as sister to those Sasquatch food, the Pedaluridae, and the Libelluloidea, which is the other group that's lost its ovipositor, has a different common ancestor. So what that tells us is that the ovipositor, or that egg-laying apparatus, has been lost twice independently. And that behavior of squirting your eggs out in a clump and tapping it on the surface of the water, it happened multiple times in the history of dragonflies. We're now starting to move towards sequencing whole genomes. This really wasn't something that we thought was ever going to be possible, but as sequencing costs have gone down, people have started, pretty much sequencing genomes as a regular day-to-day thing to do. There's many genomes for things like Drosophila, bees, ants, and wasps. For dragonflies and damselflies, we are really at the beginning here. So there's very few. Just in the last couple of years, the number of genomes have doubled or a little bit more than doubled, from 3 genomes to 8. This is, by contrast, is like over 30 or 40 Drosophila genomes alone just for that one genus. But we're getting there. So we have now quite a few, 8 genomes for odinates. And these are some of the taxa for which we have genomes. And what that allows us to do is that once you have genomes, you can actually look at gene evolution. And sometimes we know the function of particular genes. So in our lab, we're trying to look at some of the genes that are related to some of these reproductive strategies, to cold tolerance, to migration, and what have you. But the one drawback, of course, of using genomes is that you really need to have freshly collected material. But that's a shame, because humans have been fascinated with dragonflies and damselflies for a very long time, and we have a really excellent museum collection at the American Museum of Natural History, the California Academy, the British Museum, the Smithsonian. There's many, many people. The Florida State Collection of Arthropods has the largest collection of odonates in the world. And so we don't want to overlook those. We want to be able to harness information from collections. And there are ways to do that because there are certain, over time, DNA breaks down. But there are certain methods that actually work very, very well with degraded or small or DNA that's already been broken into small fragments. And so we can use those types of sequencing methods to sequence things from museums. But when we go to start doing that, it actually makes us, I would encourage us all to stop and think about who has access to the museum specimens. A lot of the specimens that we have in our museums, we have because of the history of colonialism. So we members of the Commonwealth were actually able to have a certain amount of wealth that would allow us to travel and collect things from certain regions, especially in the global South. We are actively trying to digitize collections, but that's a time-consuming process. There's a lot of people in the regions from which these dragonflies come that don't actually have access to specimens from their own country, and they don't have access to the capacity or the resources to do a lot of this genomic work. So a big part of what the grant that I have right now is focused on is capacity building in the global South to really build a community of odonatologists from regions working on specimens and species from the regions from which the researchers hail. The question I'll briefly touch on from this list here is whether or not there's DNA in any of these older samples. I mentioned before, color is a really big part of the story for dragonflies and damselflies, and humans have tried to preserve the color of dragonflies. The body color has pigments, epithelial pigments that kind of migrate up and down in the epithelial cells. And when they die, the pigments kind of drop to the bottom of the cells and they end up looking very dull. So humans have tried to preserve odonates in all sorts of chemicals before finally settling on acetone or fingernail polish remover, which works really well to preserve colors. So most specimens collected after 1970, which were preserved in acetone, actually have very intact DNA fragments that are around 600 base pairs long, perfect for doing the sequencing work that we wanted to do. But we had questions about some of the older specimens, whether or not there was usable DNA in there. Our initial work, we did some test plates. This is a paper that's just in review right now. And I wanted to give a shout out to my former undergraduate student who's now a PhD student at CUNY, Renato Nunez. He's in David Lohman's lab, and he's done something similar for looking at butterflies and using butterflies from museum collections. And in butterflies, they either stored them in ethanol, or they just dried and papered them, or they pinned them. And he looked at DNA quantity and the amount of genes they were able to sequence from butterflies that were preserved in these different methods. It's a very neat paper. It just came out, so please check it out. For our question, we only mostly had dragonflies that were either dried and put in envelopes, or dragonflies that were treated with acetone and then put in envelopes. We used samples from the Naturalis Museum, which is the acronym RMNH, which is from the Netherlands, and we used samples from the American Museum of Natural History. And we used the oldest sample that we had, I think, was from 1909, so fairly old. And as specimens were older, we got less DNA from them, as you might expect. But what was interesting is that regardless of age, we actually were still able to get quite a few loci, with quite a few genes that we were able to amplify. So the average number of loci for the American Museum samples was around 100 genes, and the average for the Naturalis Museum was around 500 genes. And we think the difference between the two museums, the reason why the Netherlands Museum had a lot more DNA and a lot more loci or genes that were recovered, we think it has a lot to do with the way that the specimens were stored. The specimens were stored with slightly different humidity levels and then slightly different, they were all stored in curators' offices for the majority of the lifespan of these specimens. But then at a certain point, we moved them to climate-controlled compactors. When we did that was slightly different between these two different museums. So we can actually look at those specimens and see where they fit in this tree of life of dragonflies and damselflies. I'm just going to zoom in here. And the specimens that were the older specimens have the year from that they were collected next to them. And you can see they're in red here. So Gauntlets cystatus was collected in 1933. We sequenced it, and it's grouping with other members of Gauntlets. So we find that congeners are grouping with congeners and conspecifics. So this is very promising that indeed we are getting specimens from these DNA from these old specimens, and the DNA is actually dragonfly DNA, and it's actually the right dragonfly DNA, which is exciting. We can also get lots of other information from these older specimens, of course. Geographical distributions is something that we've been publishing on a lot recently. And this all ties into our work looking at how dispersal capabilities affects speciation. Which is a good segue for me to mention that there are a lot of dragonflies that are really found kind of globally. And so we're not just interested in looking at deep time. We're interested in looking at the population level and looking at variation among individuals in different regions. So one of the areas that we've been sampling a lot is the Arctic. So people often think of the Arctic as being kind of habitats that look like this. But of course, during the summer months, the Arctic looks like this. So these are some photos that I took when I was in northern Sweden, in Phylogenetics. And for much of July and August, and even parts of early September, it looks like this. And this is great habitat, of course, for adults of dragonflies. The nymphs usually of any dragonfly or damselfly live from a period of time from weeks to several years as nymphs. But then the adult period, they just usually have 2 months to do all those jobs of dispersing, finding a mate, mating, and then they die. The temperatures that you find in some of these northern habitats are actually not as cold as you might imagine. And so this is actually perfectly normal to find adults flying during temperatures such as this. So we've sampled a lot north of 66 degrees, which is the Arctic Circle. And I did a lot of this work with Manpreet Kohli, who is a professor at CUNY at Baruch, and Göran Salayan, who's a professor in Halmstad in Sweden. And we had these questions about whether or not we could try, well, I guess we're in a race against time as the Arctic is ever warming, and we know that some of the habitat is being lost. We wanted to collect baseline data for the taxa that are north of the 66 degrees. There's 48 dragonflies and damselflies that are found north of the Arctic Circle, and 6 of them are actually circumpolar, which means that their populations extend Sweden, Finland, Norway, Russia, Alaska, and Canada that kind of are found around the entire kind of range, circumpolar range north of 66 degrees. So we've been heavily sampling these 6 species in particular. So as I mentioned, the nymphs can survive as sometimes multiple years as nymphs. And in the winter in northern Canada, in Sweden, in Finland, and the places I just mentioned, they can survive these winter months either as nymphs or as eggs. And some have suggested that perhaps this is a great evolutionary strategy because it's 4 to 5 months predator-free while you're either below the ice, just kind of not moving around very much, or in some cases actually frozen solid in ice. And Gordon Pritchard and others have done research where they've taken ice cores out, they've had individuals that were frozen solid, and once they thawed, the nymphs were able to kind of walk around and move around. We put data loggers ourselves up in northern Sweden, Finland, and Norway, and we actually found that the area where many of the species that we were studying were kind of hanging out during these winter months underneath the ice was, 4 degrees, 3 degrees. It was actually above 0 in this very bottom kind of mucky layer below the ice. Part of the lakes, some of the lakes, but not all, that we are doing our sampling in our permafrost bottom lakes. Permafrost is this layer of permanently frozen ice that underlays a lot of these Arctic habitats. There's usually an active layer that kind of freezes and thaws every year, and then there's this permanently frozen layer. And in many cases, as the permanently frozen layer or active layer is lost due to global warming, the entire lake is lost because often there's no bedrock, there's nothing kind of holding the water there other than this frozen layer. So when the frozen layer melts, the water just kind of seeps into the ground. Of course, there's the added negative effect that the permafrost actually has a lot of stored carbon, and so carbon is also released, further exacerbating climate change. So we see hummocks of this permafrost in things like Palsa Myers. So as we see this loss of Arctic habitat, we want to try and document these species kind of quickly. And the one that I'm going to mention is Somatachlora salbergi. This is an emerald. They're called emeralds because they have bright green eyes. This is what the nymph looks like. And traditionally, people thought they were only in these very, very remote Arctic locations in Canada and then Fennoscandia, which is Finland, Norway, and Sweden. So we kind of divided and conquered. Manpreet went to the Yukon, and Joran and I went to Fennoscandia to do some sampling. So in Fennoscandia, the pools were in September hovering around 0 to -1 degrees Celsius. And we went and we collected these nymphs. We found Somatachlora. We were pretty excited to find them. We went to several sample sites as far north as to the border of Russia. And we found Somatachlora salbergi. And then we turned around and we started driving south down through Finland. And we stopped and had lunch and we saw a lake and we decided to sample there far further south than we would have ever expected to find Somatachlora salbergi, but it was there. So then we kept driving further south and we saw some more lakes and we stopped and we sampled and it was there. So it seemed like what we found from our study is that Somatachlora salbergi was found, yes, in these Arctic habitats north of 66 degrees, but it was also found much further south than it had ever been recovered. So for Manpreet, she went to the Arctic sites in the Yukon, and she drove up and down the Dempster Highway, and she did not find them. She found other Somatachlora, but she did not find Somatachlora salbergi. And she wondered whether perhaps they were being outcompeted by species of Somatachlora that were now moving into Arctic habitats as they've been changing. Eventually, she found Somatachlora salbergi, but much more further south, much further south than had been previously recorded. So we took the samples that we collected from these different locations, as well as a few from museums, and we sequenced the cytochrome oxidase 1, or the Co1 gene. And what we found is that they formed one single clade, or one monophyletic group, one single group of Somatachlora salbergi. We can look at what's called a haplotype network. In a haplotype network, there's circles. Circles indicate a genetic pattern. The size of the circle indicates the number of individuals that share a particular genetic pattern, and the colors tell you the region from which the individuals came. And you can see that all of the samples that we had, regardless of whether they were from any of these countries across their range, they all shared one main genetic pattern with there's 3 additional genetic patterns from the Yukon. We also found that one of the specimens that was found in Japan, which was thought to be a different species, is actually Somatachlora salbergi. So we have a new location now for Somatachlora salbergi. So it's possible that Somatachlora salbergi are expanding their ranges southward, but it's also possible that perhaps they were there all along and people just didn't think to look for them. So this is an example of a taxon that probably we're going to lose in my lifetime. We think that this population is decreasing, but I thought I would kind of end on a slightly brighter note on something that might be a winner. So this is the blue dasher, or Pachydipplex longipennis, and in this game of climate change, it actually is doing really well. So Pachydipplex longipennis, this is work that I did in collaboration with an undergraduate in our lab, Ellie Gamet. But she had this hypothesis that perhaps Pachydipplex was expanding their range because they're able to actually survive in water features in people's homes, in dammed areas, even in swimming pools. And so she wondered whether these urban kind of or suburban areas allowed them to kind of pond hop into new locations. So she looked at all of the records of Pachydipplex longipennis that we have across the geographic range. It's only found in the United States and Canada. And we have a lot more records recently because of things like iNaturalist. So these are the variation in where we have found them from 1990 until 2020. And if we look just in the last 4 years when we've had iNaturalist, these orange dots indicate individual occurrence data, like where humans have observed them to be. In this, there's the colors of the map, the brighter yellowish, greenish colors are habitats that we consider to be the most suitable. And dark blue, especially darkest blue, are areas of least suitability based on climatic variables, temperature, precipitation, etc. And what she found was that in parts of their range, like in where she's actually from Las Vegas originally, it was predicted to be a highly suitable area, and there were many occurrences. So places like Salt Lake City, Provo, Las Vegas actually had high occurrence records. There were other areas that were similar geographic area, like some of the national parks in Utah, and we did not find any occurrences there. They are lacking these kind of human-made features. So part of our hypothesis that we're still looking into is that perhaps Pachydipplex longipennis, the blue dasher, is actually able to kind of thrive in our rapidly changing environment as we convert land into suburban areas. We have all of these water features. They're really able to kind of expand where females, their egg-laying habitat has increased. This is an example of a dragonfly that doesn't have an ovipositor, and it just taps its abdomen on the surface of the water to disperse its eggs. Of course, all of this is in reference to insect decline. The reason why we want to rush to collect data in the Arctic, the reason why we're interested in figuring out which species are thriving and which species we might lose is because we know that we're in a period of insect decline. So there was a paper that came out in 2017 that used biomass, the weight of insects that they had collected over a 30-year period to kind of sound the alarm about climate change, perhaps leading to insect decline. It certainly wasn't the first paper that had shown this, but it certainly got perhaps the most attention. And some criticized this paper because they said that weighing the total number of insects in an area isn't a good predictor of whether you're losing species, because the weight of a mosquito is very different from the weight of a housefly, which is very different from the weight of a butterfly. And so there was kind of a call to collect species-specific data. Well, people have done that. So certainly since that paper came out, and even before, people have really written a lot of species-specific data. And what the data seems to be telling us is that now is the time to act. We are indeed facing a period of insect decline. The rate of decline is higher than we've ever seen in human record, and we need to do something. You can go to this website. There's a screenshot of which is on the upper left-hand corner called Entogem, which actually has synthesized and collated all of the existing data sets that are out there and published papers on the area of insect decline. There's a great paper by my colleague and collaborator, David Wagner, called Death by 1,000 Cuts. It came out in 2020, and it kind of talks about the different drivers that we think might be causing this insect decline. And it's not one simple solution. Like if we got rid of law, then we would be able to save insects, because there's actually lots of little things that together have added up to cause this negative impact. So we have a big grant right now. Christy Bilal, David Wagner, Eliza Grames, Chris Elphick, and myself. And we're actually kind of synthesizing data and making predictions and hopefully suggestions on how to act so that we can perhaps lessen the rate of decline of insects. And if people want to get involved or join us, please just shoot me an email after this call. These are some photos of my kids when they were younger. They used to love to go out and collect insects. Collecting insects or flipping over logs or picking up frogs is something that I think a lot of us remember doing as kids. And our goal, one of the easiest things that we can do to really change people's perception about insects and hopefully stave off this kind of insect decline, is to just keep people fascinated in nature, keep people asking questions, and kind of treasure that natural curiosity that people seem to be innately born with. This is a picture of some insects that my kids and I made when we went to Guyana. I took them to Guyana in South America with me, and we used an old bingo box and a piece of upholstery foam, and they were able to pin their insects there. That curiosity is what we want to kind of harness in everybody, in hopes that that might change people's perspectives about insects and help us stave off their decline. And so with that, I'll thank my collaborators and funding sources. I'd like to thank my research group at the American Museum of Natural History. These are my students and postdocs, as well as my past students and postdocs. And if there's time, I'll take any questions. Thank you.
All right. Thank you so much, Dr. Ware. Let's see. Can everybody see me as well? Hopefully. Hi, everybody. I'm Gabriel Willow, the Vice President of the Linnean Society of New York. And again, thank you so much, Dr. Ware, for being with us tonight and for that fascinating presentation. I'm a huge odonate fan myself. I'm a little rusty these days on my odonate ID, but I used to spend a lot of time chasing them around swamps and marshes and ponds and things. So folks, if you have any questions for Dr. Ware, just go ahead into the Q&A section that you'll find at the bottom of your screen, and you can type them in there. We already have a few questions coming in. Actually, before I get to those, I have to ask. My favorite dragonfly actually is the gray petal tail, and they can be found not too far outside of New York City, up at Doodletown by Bear Mountain. I see them there quite regularly. It's such a fascinating, strange, spectacular beast. Just to clarify the connection between petal tails and Sasquatch, does the presence of petal tails indicate the presence of Sasquatch?
Well, so that's the million-dollar question. So Perry Turner, who is this graduate student at Berkeley in the 70s, brilliant biological illustrator. He did some just fabulous natural history on this group, but he did veer into cryptozoology and into the study of Sasquatch while doing this thesis work, which he presented, and he got his PhD. But his work is at the Smithsonian. Long story short, the contents of his storage locker ended up being sold for auction, and anyways, there was like type specimens that were in there, and the Smithsonian got them. There's been a bunch of stuff that's been written about them, which I can happy to share later. So I've read it, and indeed, he does say that the primary diet of Sasquatch are petal tails. So where there are petal tails, one would find Sasquatch. That's what he says. The distribution, just for the audience, there's the petal tails in North America. There's the East Coast, which is Tacopteryx. It's a single monotypic genus. And then the West Coast is Tanipteryx, which is a single monotypic genus. So he was studying the one on the West Coast, which makes sense why he was talking about Sasquatch. There's also one in Chile called Phoenes, and then the rest are in Australia. And as far as I know, there's no Yeti or Sasquatch records in either of those places. So,
well, correlation is not causation, but you heard it here first, folks. If you're up at Doodletown, where also it's well known for its cerulean warblers and some other birds, be careful. There's rattlesnakes there, but there's possibly also Sasquatch or Bigfoot, apparently. That is a fascinating connection. And I wasn't sure. I was like, did Jessica just throw that in there to make sure we're still paying attention? Or no, this is a real guy, huh?
Yeah, it's a real guy. It's a shame. Like I said, he disappeared and hasn't been seen from. I have a colleague, Chris Beatty, who's been really interested in this person and knows an awful lot more about him. And I was telling him you should make a movie about it because you could.
Yes. Or a future Linnean presentation, perhaps.
Yeah. Oh, yeah. He would be a great speaker because the story itself is really very fascinating. When you have these really enigmatic taxa, I suppose that maybe attracts someone who's enigmatic themselves.
Wow. Okay. Well, I just could talk about the Sasquatch dragonfly. I mean, first of all, just like dragonflies don't seem like a huge nutrient source for Sasquatch, but anyway, we'll save that for the future in-depth investigation into the Sasquatch petal tail connection. Let's see here. Someone in the comments gives a shout out saying those interested in odonates should join the Dragonfly Society of the Americas. Only $15 a year. That's a great tip. Right on. Let's see here. What are major, aside from American kestrels and evidently Sasquatch, what are some major predators of dragonflies? Is a question we have here.
Oh, that's a good question. So really, I mean, a lot of birds, insectivorous birds, eat dragonflies. There's lots of birds that eat dragonflies on stopovers. So there was actually, I have a colleague in Rutgers in Newark that was studying birds at stopovers in Newark, and they're routinely eating dragonflies on a migratory journey. Lizards, frogs. I've certainly personally observed this as have colleagues. Fish jumping out of the water to catch dragonflies and damselflies. Where I'm thinking of, it was mostly rock bass and things like that. But there certainly are a variety of vertebrates that consume them. Amanda was the PhD student. She was actually really interested in frogs eating particular damselflies because there was a damselfly called RJ apicalis that actually changes its color. So it can be bright blue or it can be dark purple, and males can actually change their color in a matter of a few seconds. She noticed, so she actually built a giant enclosure out of doors, that was over they like low-tech habitats, like a flowing water, that had frogs and other things in it. And when males and females mate with each other, the males, as soon as they had done the sperm transfer, changed their color to being a dark purple color. And through her work, she found that the most likely hypothesis that was supported was that it was predators like frogs and birds that were driving this color change, because it allowed them to be less visible in the habitat while they were doing this mating experience. And she has a lot of funny stories about all the frogs that ate her damselflies. And she also fed them to chickens. And she painted them different colors to see whether or not chickens, colors that were colors that chickens could see, to see whether or not, did the predations, the strike pattern, and it did. So I think that, these vertebrate predators certainly have impacted color. We know that they've impacted flight style. So before the rise of modern birds, those dragonflies and damselflies have very different wing venation, and their wings tend to be very, very densely veined, which we know is probably related to having a very stiff wing. You can measure flexural stiffness, you can measure how stiff something is, and denser wing venation tends to make a stiffer wing. And we think that they were actually rather poor flyers. You see the rise of modern birds, and there's a bunch of different adaptations after which those arose. They are not lost in subsequent odonates, and we think that they're related to maneuverability and escape from some of these fast-flying predators.
That's so cool, especially considering that they've been around for, the damselflies was 225 million years, and how much was it for dragonflies? How long are we talking again?
The same for Anisoptera, yeah. Common ancestor of the two of odonates being 250-ish.
Mm-hmm. It's amazing that they're still it's just evolution is amazing that they're still coming up with new solutions and new responses to pressure from predators. I guess that would be, we're talking modern birds, that would be more than 100 million years into odonate evolution. That's neat. They can still try new things and adapt. Speaking of wingvenation, so there was a wingvenation question. Does the venation in dragonflies differ at all during its lifetime?
No, so it doesn't. So the nymphs they develop in freshwater, and the wings develop while they're nymphs. They're kind of folded and scrunched up in these what we call wing pads. When they emerge to the adult stage from the water, they kind of unfurl their wings, and then their wings stay ever thus. So there's the wings stay exactly as they were when they were kind of first forming in the water. So the venation pattern doesn't change. The only thing that changes really is over time, the wings get really battered. So you can tell the age of a dragonfly by looking at the edge of the wing. If it's really tattered and worn, then usually it's an old dragonfly that's probably close to the end of its time. And if they're very shiny and without a lot of tatters in there, what we call tenebral. And those are new, more newly emerged dragonflies. As an aside for Rosemary, this is actually really great because we can use those wings to tell us something about migration. The wings are made up of hydrogen that they get from the water, the H2O. So we can actually measure the weight of hydrogen, which varies along a latitudinal and longitudinal gradient. And that tells me if I catch a dragonfly in Namibia, I can measure the H2O, the hydrogen signature in its wings, and I can tell whether that dragonfly emerged from water in Namibia or if it emerged in Canada or Guyana or Peru. And so this is kind of a neat feature of dragonflies that allows us to kind of remotely track their movement.
That's so cool. I think it's interesting how few people realize dragonflies are migratory. I mean, like monarchs get so much press, and it's like dragonflies are doing just equal feats. I guess it's that they don't all winter in these huge conglomerations. But yeah, I mean, they're going to the Caribbean, or like I guess we don't even know where a lot of them are. The picture's becoming clearer, but some of it's still unknown where they migrate to, right?
That's true. There's less than 20 things that we consider to be true migrants, and less than 10 that we consider that we know that they exactly what they're, or we have ideas about what their route might be. But a lot of them, we just know they leave from a certain area, but we don't necessarily where they go. We just know that they go from here, but then the next step we don't really know. Pantella, which is the one that's cosmopolitan, we've done some work on that one in my lab. We know that it's global because we can look at its genetics, and we see that there's gene flow across the range, but we can also look at these wings and look at the hydrogen. We know that they're kind of moving around. And they've done some of that for some of the achienids that are migratory and the libellulids or skimmers that are migratory. But for a lot of them, it's just, it takes someone who just is fascinated to actually, go through and do all of that things. And there just hasn't been as many people doing those types of genetics or.
Yeah. Very cool. I have a couple questions about sample collection. One attendee asks, when you collect samples, how many are you actually collecting? And then somebody else asked, is there any thought of regulation, or does sampling of insects or specimen collection have any impact from a conservation standpoint, given that many insects are declining?
Yeah, that is a good question. And because we don't know a lot about what population sizes are in any given area, it's hard for an entomologist to say, oh, collecting doesn't make a difference. People have written those papers, but it usually is only relevant to that particular species that they were collecting in that particular region. So, Culex, which is a type of mosquito that we have here in New Jersey, we know those populations are really big. Sure, collect 100, collect 1,000. I don't care. There's still going to be a lot more because we know those population estimates. For the majority of insects, we don't know their population sizes. So what we usually do in my lab is actually we're very cautious about over collecting. So we usually only try to collect, a single or a couple of individuals. Sometimes you want to have more than one because what you collect, you go back to the lab and you might realize, oh, it's a different species, or maybe it's a new species. So sometimes you want to have more than one, but I try not to collect, any more than we would ever need for the study that we're trying to do. We want to try and have good museum collections, though, because museum collections are kind of our baseline data. They are kind of the snapshot that tells us what was found where. And thanks to the people who collected in the 1920s or in the early 1900s, we now know Pachydiplux used to be here, and now it's in this slightly different area. So there is some value in doing collections, but collecting exhaustively in an area, I think, is really risky for groups like insects where we really don't know very much about population size. So that's that. I guess my short answer is just that I feel particularly scared about this. The more one reads about insect decline, the more one feels kind of agita or anxiety and angst, ennui, because the fate of humanity is so intertwined with insects surviving. If insects were to go extinct, and I realize there's lots and lots of species, as we just talked about, so they aren't all going to go extinct. So there's going to be some winners and losers. But if we lose certain key species, colleagues like Doug Tallamy has estimated humans might live 3 months. I would like our species to survive. I have children. I would like them to be able to survive to adulthood. So for me, I feel very invested in preventing insect decline. And so over collecting, I don't want to do. But at the same time, as a scientist, I know that there are some things that we actually need to collect so that we can understand patterns and then advocate for science policy, that the people who are actually making decisions, that they do something that would actually protect insects. Because for me collecting 5 fewer dragonflies has far less impact than me getting the president to do something that's going to affect broad strokes climate change, right? So
yeah. Oh yeah, pretty cool that you have the president's ear. Congratulations on that award. That's pretty neat. Yeah, that tends to be my take as well, especially I guess with insects, impression is that they're all super abundant, but that's not necessarily the case. I guess with dragonflies or odonates, some of them are probably quite localized, right? Are there certain odonates that have just that are known to have really tiny populations or tiny ranges? Like, there's some birds that are maybe found on one mountain or something. Is there any odonates that are only found in like one lake or something like that?
Yeah, so we do see there's, of course, it varies geographically. So there are some areas where there's a lot of endemism and other areas where there are other species that are very cosmopolitan. There are some that are really restricted to certain elevational kind of gradients. And there's a fellow who's a colleague from Colombia, Cornelio Bota. He's done some work looking at kind of elevational species, clines, and odonates. And it's terrible news, right? Because there are some of them that have a very narrow niche space, right? They need a certain climate that's very, very high elevation. And as that climate is changing, their only fate is death, right? Their only fate is extinction. So yeah, there definitely is that same pattern in odonates as what you're seeing in birds for sure, where there's some cosmopolitan and some that are very specific to a given niche space or are very endemic to a certain region.
Yeah, well, like that petal tail, I mean, I think they're very sparsely distributed to certain spots, like Doodletown, where oh wow, I see them every time I'm there, but I've never seen them anywhere else. And my understanding is I think in New York State it's a threatened species, maybe. Yeah. Well, that's nice to hear that you're so conscientious about your collecting, but there's no real legal protections, right? Like anybody could just go out with a net and catch a dragonfly and pin it in their specimen box or whatever if they were so inclined, right?
Yeah, well, I would say don't pin it. You should please put it in an envelope because dragonflies fall off when they die, and it's a feature of their neck and the way that their head isn't really attached to their neck. It's just resting with this special Velcro system. And when they die, the Velcro unclasps and their heads fall off. So you put it in an envelope so that way the head stays with the thing. But there's no restrictions here in North America. In Europe, yes, you cannot collect dragonflies in most of Europe.
Interesting.
Like at all. And of course, in other places, there's very strict permitting. But some have really pushed back against that because what that means is that the types of data that my colleagues have to work with in Europe for doing assessment is actually very different data. It's just based on site, sometimes from far, because you can't even use that in some places to do it. So it can be, there's a lot of debate, I would say, about what the best method is. But here you can collect them unless it's listed. There are some dragonflies and damselflies like emerald that you cannot collect. You ought to collect things that are of concern. The petal tail is also a concern in New Jersey. We haven't seen it since I think 2001 was the last official record, although there's reports. I've seen one. So there are reports of another population that's in the kind of central south New Jersey, and people have been talking about it. And I went there and I saw one, but that's not somewhere that you want to collect. There's so few.
Yeah, is this near the Pine Barrens? Like, could there be like a no, it's not the chupacabra, because it was in the Jersey Devil connection, maybe?
Oh, yeah. I don't know if you eat one if you become a Jersey Devil.
Yeah, you got to look into that. I think that's another area of research. You must have been pretty excited to find one too. It's such a magnificent creature. Now here's a slightly lighter question. This is fun. There's a whole story here actually. Leah says, I live on an island 30 miles north of Seattle. This summer, a damselfly landed on my nose as I was floating in a local lake and stayed for a while, trying my cheek and chin, always returning to my nose. He or she took off twice and chased an approaching damselfly and then returned to my nose. Can you comment on this behavior?
Yes. So Leah, I think that means that you had a male and he had made your nose a territory. So there are some dragonflies and damselflies are territorial. They're not all of them are, but the ones that are territorial will choose a territory and then they will defend other males in the area and chase them off. The territory is usually near water, so it makes sense if you were floating in a local lake. And the reason why they're doing that is they are basically saying, like, as far as I can see, the females in that area, I'm going to mate with them. And if other males come in, they'll chase them away so that way they can go back to the territory and then find the females to mate with them. As an aside, there are sneaky males while they're off chasing the males away to get them out of the territory. Sneaky males will just come in and mate with the females. But that's my guess, is that you had a male and the male had chosen your nose. Was it your nose? I think it was or your cheek,
It was
whatever.
nose, yes. It was the preferred perch. Or possibly Leah floated into his existing territory, right? And then she was just a convenient perch. And the nose perhaps was the highest point if she was floating on her back.
Mm-hmm.
Yeah. I wonder if the dragonfly cares if it's a log or a Leah in this case. Or
Yeah, I don't. I know people that do this. Like they'll just stay like this for a while, and eventually this becomes the perch. Like put your finger out. So if you're fishing, I have a son who likes to go fishing a lot, and his fishing rod is always a great perch when he's standing in the water. Yeah.
Not to say that you're not special, Leah. I'm sure the damselfly recognized something inherent about you that was cool and to perch on your nose. Let's see. There's a couple of good related questions. It's actually a series of questions. Actually, I was wondering this myself. How long is the average lifespan for an odonate? And are they solo living insects, or are they found in swarms or flocks or with family members?
So it depends on the species. There are the shortest lived are things like Pentella, the global wanderer, which is a migratory one. They develop as nymphs in 6 weeks in the water. They live for 2 months as an adult. But there are really long-lived ones that take like 5 or 6 years in the nymph stage in the water, and then they emerge for 2 months. So the adult stage never really varies. The adult stage is like 2 to 3 months. I think I have a colleague who did some mark recapture work in Colombia, in the Amazon side of the Andes, and I think the longest was maybe 4 months, but like that's pushing it, right? It's a really short window. The adult. What varies is this juvenile stage, and so the juvenile stage, as I say, between 6 months to 5 or 6 years as a juvenile.
Yeah. I think that's the case for so many insects and their longevity. Like mayflies have this sort of perception of like, oh, they only live a day or whatever, but as nymphs, they're living a couple of years or something. So a lot of insects, I guess, have these longer nymphal stages that get kind of dismissed, and then the adult stages are a bit more ephemeral. Yeah.
About their solitariness. So mayflies are a good example where because they all kind of emerge at the same time, they're aggregated because the timing is important. If the adult's only going to be around for 24 to 48 hours, you want to make sure you all arrive at the same time to the party. Otherwise, you're not going to find a mate. But for odonates, it's not really like that. So for odonates, they're really very solitary. So they sometimes will have groups of females in a given territory, which they call kind of lacking behavior. But it's not that they're looking for each other. It's that they've all gone to this one water source to lay their eggs. So we don't really think of them as being groups or family groups, and in fact, it's brutal because if a female lays a clutch of eggs and then she lays another clutch later on, the first clutch will eat the second clutch. Like there's not a lot of family love, there's a lot of desire for sibling selection for you to try and favor your siblings. They'll just indiscriminately.
But you said that's if she lays another clutch later, but within a single clutch, did they somehow avoid eating each other?
Nope, birth order matters. No. So which one hatches first, it's free game to eat anything else.
Wow. Yeah, that's pretty rough. Now here's a somewhat related question. Is anything known about dragonfly intelligence compared to, say, fruit flies, where it has been studied?
Well, we don't know very much about what intelligence means for insects, I would say, and certainly not for odonates. What we know is that we know a little bit about their brains. We know that they can do some slightly sophisticated styles of hunting. So they do what's called interception predation. So like a lion would, they look for their prey. Say they're going to catch a fly, right? They look at where the fly is flying and they go to where the fly will be in 10 seconds or whatever, and they cut it off at the pass, right? They intercept it. You don't follow the fly like a dummy, follow the fly in its path. They go to where the fly will be and they intercept it. And some have said, I have a couple of colleagues that study this, and they consider that to be rather sophisticated. We know something about the number of neurons in the brain that we think are related to this. And it's not that many. There's not a large number, so it does seem like that seems sophisticated. We know that they have the ability to perceive images in a slightly sophisticated way. Does that mean that they're smart? I don't know. So I guess depending on I guess it depends on how you define intelligence. But they seem to be able to recognize things in the environment, like perches, like vegetation. They're able to change their speed dramatically while they're flying. They're able to, like I said, perceive each other and they're able to communicate with each other using signaling. So sometimes it's color, sometimes there's color on the wings that they can actually flash in a certain pattern. Does that mean that they're smart? I don't know. So they do behaviors that some could say are sophisticated, but whether that means that they're intelligent, I don't know.
Oh, that's a wonderful answer. Yeah, I think so many things that we consider intelligence just have a type of social intelligence that's human-like, and so when we recognize it in corvids or cetaceans or something, it's just because it reminds us of ourselves on a lot of levels. Like I think it's a really good point to like move away from even saying that intelligence is objectively a thing. Really, yeah, very interesting. I think we have to wrap it up pretty soon. There's a bunch of good questions here, but quick one is that I was wondering too, so I'm a little biased here, is with the migratory dragonflies, because you mentioned they only live like 2-3 months, so that got me thinking, do the same individuals migrate somewhere warmer and then migrate back, or is it a second generation making the return trip, or do they not return at all? Or
that's a good question. So they don't really go for warmer or colder areas. It's not like birds go to new areas where there's water. So they take the ones that are migratory, and we don't know all of them where they go, but for the ones that we do know, for like the global wanderer, it uses kind of winds around the equator, the intertropical convergence zone, these series of winds that go around the equator, and they basically just, when they're ready, they pick up their feet and they get carried around, and those winds kind of follow the rains, right? And so they kind of, as a rainy season just is finishing, they drop down, lay their eggs in all those temporary pools of water that have kind of accumulated, and then pick up their feet and then go to the next spot. So it's some have said it's a strategy for females, right, where they can lay their eggs in these multiple, disperse their clutches over, the hedge their beds. Don't put all your eggs in one basket. There's a metaphor coming there. So that's what we think that they're doing, and it usually individual that can, as far as we know, there are single individuals that can travel 11,000 kilometers, which is like farther than monarchs, and that's a pretty big one. That's a good thing. But of course, they stop on islands. A very famous stopover is in the Maldives, in the Indian Ocean, for example. We don't know an awful lot about the island populations for any of the islands for Pantala, which is this genus I'm talking about. With the Maldives, we know an awful lot about, because humans live in the Maldives, and they've been making observations like, hey, every year around this time, these giant groups of these particular dragonflies arrive. And so when they stop over, they lay their eggs. And because the development time is like 6 weeks, of course, some of those individuals end up joining this larger swarm, right? So then you could, in theory, have multiple cohorts in one large group of Pantala that are flying around, I guess.
Fascinating. Wow. Yeah, I sort of fundamentally misunderstood dragonfly migration. I think I'm kind of more bird guy. I think I was thinking of it as a bird-like migration. I mean, the monarch migration is sort of bird-like in that regard. So that's really interesting. It's just sort of a dispersal. Yeah, because sometimes I'm trying to remember when it is. I feel like it's like late summer, early fall, maybe September, in like Prospect Park, for example, in Brooklyn, over the meadows. There'll just be hundreds and hundreds, maybe thousands of dragonflies swarming around. And I always thought of it as a like southbound migration, but maybe I was wrong this whole time.
Yeah, that's often Pantala. What you see, they're often over like that, over meadows. They're building up their fat stores, is what they're doing when you do that. They're just consuming aerial plankton. They have spines in their legs. They're just jamming food in, and then they're off. We don't know this because, of course, we don't have enough data to really say this. But anecdotally, and from our preliminary data, those individuals would be then continuing on to the next part of their journey. So they're building up their stores. When they lose their fat stores, they just drop from the sky and they're dead. So the goal, it would be to build up its fat stores. You would think selection would act on you building up your fat stores before flying out over open water, because ocean is death for them, because salt water is death.
Yeah, very cool. It's neat to get to witness a little piece of that process. Last question, I think. Can you, and sort of end on a conservation-oriented note, can you say something about the significance of insecticides in comparison with other influences on the decline of insects and odonates?
Well, certainly insecticides for insects in general have a negative impact. There are some insecticides that we feel like we need to use to preserve food security or to prevent vector-borne illnesses and, ultimately human survival. We in the global north can't really say you can't use certain insecticides for things like locusts that can cause great human suffering if they destroy entire crops in entire countries. So we're kind of walking this fine line between using the chemistry that we have and preserving insects. Having said that, there are certain types of insecticides, because I shouldn't say specific brand names, but there are certain types of insecticides that we know have a negative impact because they have a lot of non-targeted impacts. Not a lot of people have studied the impact of insecticides on dragonflies in particular, but of course, dragonflies are indiscriminately eating insects out in the environment. So any of those insects that have visited flowers, and a lot of them are herbivores or pollinators, if they have sequestered or have the impacts of any of those insecticides, that could negatively impact in dragonflies. But of course, the bigger issue is just that as this trophic level is lost, as herbivores and pollinators are lost, the main diet of dragonflies and damselflies are lost, right? So they just have less food around to eat. If the populations of butterflies or what have you get smaller, that's a great food item normally for a dragonfly or a damselfly. So we don't know very much about. A lot of times people will put we use methods called integrated pest management, where we try and use biological control agents or non-chemical methods to try and control insects. And we have some really good ones, like Bacillus thuringiensis is a bacteria that kills insects, and people will use them often in water. They will, especially in New Jersey, they do this all the time in New York, New Jersey, will put it in water to lessen the number of black flies and to lessen the number of mosquitoes because they also have fresh water. We do not know what that does to odonates. So we don't know what that's actually the diet of the nymphs. We don't know if it actually impacts the nymphs themselves. So there's like a lot of use of insecticides without necessarily really understanding the impact of this kind of top predator in a lot of these systems. So we don't know, and I guess I would say for odonates, we just don't really know enough information. But my suggestion would be that it probably will have a negative impact, either directly for some of these non-target effects, but then also just because it lessens the amount of food that's available for odonates to eat.
Sure. Yeah, and I guess pesticide runoff would make its way to a lot of pools and ponds that odonates utilize as well, and so maybe you would have some higher concentrations there. I guess now that I think about it, that's a bit of a depressing note to end on, but at least there's probably some conservation-related thing there.
And I will say, like 6,500 species. I'll spin it. There's 6,500 species, and they're not all the same. So, like the blue dasher that I mentioned, can develop in a chlorinated swimming pool just fine. So there are some will be winners that will still serve. There are things that are not very sensitive to these pollutants. The problem is that we're losing a lot of these really cool, rare, very specific taxa, and that's a bummer. But there are going to be still things. So there's always going to be a blue dasher type thing, a blue pachydiphlex. For every five that we lose, there's a pachydiphlex that loves this altered habitat.
Yeah. Well, thank you so much. Yeah, it's really interesting to see how these things play out over time, and it's so interesting too the way you tied in climate change and the Arctic research, and it's just absolutely fascinating. So it was really wonderful to have you, Dr. Ware. Thank you so much for this a wonderful presentation and conversation.
Having me.
Yes, and I'll just tell everybody that next month's presentation will be wildlife movement and infectious diseases, so very topical, with Meredith Van Acker. So I hope everybody can join next month. And thank you once again, Dr. Ware, so much. And with that, I will officially bring tonight's meeting to a close. Thank you all so much, everybody, for joining us tonight for the Linnaean Society's monthly presentation. Hope to see you all again next month.
Thank you.
Have a great night.